Okay, today we are going to finish suffix arrays and start patterns, profiles, and we are going to see hidden Markov models today to represent multiple sequence of protein, family of protein sequences. Suffix arrays is a variation of suffix trees, but the only thing that is common between suffix trees and suffix arrays is that they both use the same idea of if a pattern exists in a string, it must be a prefix of a suffix, okay? That's the only thing that is common. Other than that, the data structure, the construction, even the searching algorithm is completely different. They do not share anything common apart from the fact that they use this observation. A pattern has to be a prefix of a suffix if it exists. So it was introduced in 1993. More space efficient than suffix trees. All we need actually is an array of integers, okay, of length n. If we have a suffix of, actually if we have a string of size n, all we need is an array of integers of length n. All we do is we store in this array, okay? So this array has indices from 0, 1, 2, 3, n minus 1. Let's say n, which has the dollar one is at the end, okay? And what we have is in this array of size n plus 1, including the dollar character at the end, we store our suffixes. Our n plus 1 suffixes. And we store them in such a way that we sort them in this array. These suffixes are sorted as if they would appear in a dictionary, which is called a lexicographic order. Okay, so we sort these suffixes as if you put them in a dictionary. If you put your suffixes in a dictionary, how would they appear in that dictionary? You sort them in this array, and this is your suffix array. That's it. That's simple. So any in-place sort, I mean, you can use any sorting algorithm to sort your suffixes in an array like this. You can use quicksort. So the construction time of suffix array is O(n) log n. However, we are going to see the search time is not as simple. The searching is not as simple as the suffix tree. The search in suffix tree was in linear time. However, to search a pattern in this suffix array, we are going to need log n time. We are going to use binary search. We will not. So this size of the string is going to be effective when doing the search. Okay, so a suffix array for a string x of length m is an array of size m that specifies the lexicographic ordering of the suffixes. So it's like this. This is an example string, ACAAA CATAT $. Okay, so this is, come on. Yeah, I hit this. I hate these interruptions, I mean, from the operating system. Okay, now, this is the example suffix array for the string, including the $sign. It has 11 suffixes, and these 11 suffixes are stored in this array in sorted order as they would appear in a dictionary. As you can see, the first thing you would appear in the dictionary is this triple. This is a suffix that starts with triple A. Okay, AAA. That is how you would put that in a dictionary. And a good thing is, the good thing here is we don't need to actually store the actual suffixes here again. Similarly, all we need here in this array, this is just for demonstration, okay? These suffixes in the middle is just for demonstration. The actual suffix array does not need to store these suffixes. It only stores, it's an integer array. It just stores these numbers, 3, 4, 1, 5, 7, 9, 2, 6, 8, 10, and 11. This is the suffix array for this string, which just has these numbers. And these numbers, they represent suffixes. So this 3 indicates that it's the suffix which starts at the third letter, third character of the string. A, C, A. A is the third character. So here our suffix indices start from 1, so, and this is the third suffix. The first suffix ACA comes at the second as the third here in this array. So this is our suffix array. Now, if we have this sorted array of suffixes, now the search becomes just like binary search. If you have something sorted, then what we are going to be looking for is we are going to be looking for a pattern. And imagine we are looking for a pattern like CAA or CAT. Or try to search for something that doesn't exist, CAG, okay? Let's try to search for a pattern CAG. The first thing we need to do is find out all the suffixes, where are the suffixes that start with C in this array. We need to inspect this array and see where are the suffixes which start with a C, because our pattern starts with a C. So we need to a binary search is implemented like this. Since we have this from 0 to 10, we divide this 10 by 2 and check, which is 5. So the one at the 5th index is A. What does this tell us? It tells us that the first half of the array does not contain any suffix that is related to our search. Okay, so because it's the first half of the array, since we hit it and in the middle we hit at this A, which means that there are no suffixes that can start with a C at the beginning part of the array, because they are sorted. So I can make sure this is true because I sorted them. So here this 5th suffix, it starts with an A, and I'm looking for suffixes that start with a C. So what I do is I just throw out the first part of the array. I don't need to do search here. So I reduce my search by taking half of my dictionary out, and I start conducting my search here. So between 5 and 10, the middle is this 8. So it's T. Again, T is a letter which comes after C. So the suffixes I'm looking for cannot be below this point, so it's somewhere here. So we search a range like this, each time dividing the search part of the array by 2. That's why it's called binary search. But here, it's a little bit different than binary search, because we are not looking for an exact number, whether it occurs or not. We are looking for an interval here. Here we are going to do an interval search during this binary search, because at the end of this binary search, I want to continue my search after matching the C, after matching the first character of the pattern with my suffix. To continue with the second character, I want to conduct my search between the 6 and 7 by using these two indices, okay? So I'm going to start an interval. My interval at the beginning will be the beginning of the array and end of the array. And I'm going to each time shorten this interval of my search until I can find my pattern exactly in this dictionary. Okay. You could also do it in this dictionary. You can do. You can apply any algorithm that can do fast dictionary search. Okay. So our goal is to conduct a fast dictionary search here. If you're searching for a pattern CAT, is there anything in this dictionary that starts with CAT? This is what you want to find out fast. And a simpler algorithm would be just to go over all the dictionary entries, okay? But this is not what you do in real life, okay? In real life, for example, if you're searching for a word like bioinformatics, do you go over all the words that start with an A? No, you immediately go to B section, okay? The dictionary is divided into sections that you can conduct. Maybe you can apply a similar idea here by indexing your dictionary based on the first letters. But then what are you going to do for the second letters? I mean, when you're in the B section, imagine what you're doing when you're looking at the B section of the dictionary. You don't go through all the words one by one. What you do is you skim through fast. You, for example, want to find out, you look for second words. How this B section, there's no explicit arrangement of the B section, but you actually apply a binary search by yourself in that point. You try to, for example, bioinformatics. The second letter is I. So you don't immediately go and look at the beginning part of the B part, because there are going to be words like which the second letter is A, okay, B-A. So like base or other things, all the words that start, in which the second letter is A, is going to be in the beginning of the B part of the dictionary. So you just imagine, okay, I should be somewhere in the middle. So you go look in the middle, and you by doing a binary search, you very fast locate the word bioinformatics. So this is something similar. The search is conducted similarly in suffix arrays. So the construction can be done m log m. As I said, all we do is we sort the array suffixes. Any sorting algorithm, you can even do quick sort to do this lexicographic ordering if you define how to compare two strings lexicographically fast. Okay, if you can compare, just like as you're comparing numbers. If you can immediately find that 5 is greater than 1, if you can do the same thing for given two strings, if you can say that which one is greater than other lexicographically fast, you can do it in OM log m time by using a regular sorting algorithm. And you can also do something like an insertion sort. Insertion sort is worse in time complexity. It takes OM square time to do the sorting. But as I said, a regular quick sort or other, you can do merge sort, which will finish in m log m time. And as you can see, the suffix array is very space efficient. You just have an integer array of a size n. You don't need any pointers. The suffix tree, you have a more complicated structure. You have nodes. Those nodes have children. You are going to need space for that. Even if it's, again, asymptotic time complexity, in big O notation, it's ON. Suffix trees also require ON storage. However, suffix arrays, this ON, there are no other constants in front of the n. Okay, it's purely you just need an integer array of size n, and that's it. And for a query, to search a query, as I said, we do a binary search, and that binary search takes m log n time. And here is the size of the database. Okay, so here this query, searching your word in suffix arrays takes into account the dictionary size. If you have a very large string, it's going to take longer time, but at the log of that dictionary. So even that's going to be a very small number. Even if this n is large, log n is going to reduce it to a small number. Okay, binary search is very fast. So here's the pseudocode to search for a pattern in suffix arrays. So this function find. It takes a parameter pattern p and a suffix array a. It stars two indices that shows the low and high point of our search. So we are going to, this low and high are going to start at the beginning of the array and end of the array. And each time by doing binary search, we are going to increase this low and decrease this high until we can find our pattern exactly. Or if we cannot find the pattern in the array, it means that our pattern does not exist in the string. Okay, so for all the characters of pattern P, so starting from the first character, we are going to search whether this first character, we are going to do a binary search for two x and y locations for this character in the suffix tree. And we are going to reset low and high based on this character search. And we are going to continue this search again. Actually, this can be a recursive function, this binary search. And we return all the suffixes between this low and high at the end. Actually, in this for loop, this is not a recursive function. Within this for loop, we conduct this search until we go all the way to the end of the characters of pattern P. If we are done at the end, if low and high are equal, then it means that, we haven't found our, or low is greater than high, it means that we haven't found our pattern. Otherwise, all the suffixes that contain our pattern as a prefix is returned as a result. So it returns all the occurrences of the pattern in the string. So for example, if we had multiple suffixes in this tree, just like this, if you're looking for AA, if we were searching for the pattern AA, here we would return both this suffix and this suffix, because both of these suffixes contain AA as a prefix, and these are the two occurrences of AA in the string, one of them here, the other here. Okay, so there are two overlapping occurrences of AA in the string, and this is indicated by these two suffixes. So are there any questions about suffix arrays and suffix array search? Okay, so I assume it's clear. Yes, there's one question.
Multiple level indexing for faster search in the arrays?
Yeah, we can do that. As in for, but it's what happens is that you need to, how many, what is the level of multiplicative index are you going to go? How deep you want to go? Oh, okay, then. Yeah, but it may be very space consuming based on the size of your string. What you can do is, you may also do something based on the expected pattern length. Because you don't do this search deeper than the pattern length. So if you can have an assumption like this, or if you do not allow to search for patterns of longer than a certain size. For example, I build a dictionary and I allow for pattern search only for patterns less than or equal to 10 characters. If you can do that, then you can build an index of 10 levels for each different character, which is going to speed up your search. You can even use some kind of, hash tables can also be used in suffix arrays. Imagine this suffix array was not a sorted array, but you just put your suffixes in a hash, okay? And your search time would be 01, okay? So this is also, I'm sure somebody must have done this suffix hashes. So you should, if you're looking for this, just search for on the internet whether somebody had done a suffix hash table. A suffix hash table would be much wiser choice, because hash tables offer search time of 01, and that could work. Just put your suffixes in a hash table and do the search like that at the end. Okay, are there any other questions? The biggest disadvantage of suffix arrays is that, or suffix trees, they cannot do, they only do exact similarity search, like exact search. They cannot, if there's a certain slight mismatch in your pattern, one nucleotide mismatch, you will not be able to find it either in suffix arrays or suffix trees. But there are extensions to that. And I'm not going to cover those extensions in this lecture, but just to show you that if you want to search non-exact similarities, there are extensions of suffix trees and suffix arrays for this purpose. Here's an example to search is in Mississippi. We first start with letter I, and we find that the letter I occurs by using binary search, we find that letter I occurs within the indices 0, 2. So the pattern should be within these indices. So actually 0 to 3, all the suffixes that start with I. And the second letter is S, and S occurs in indices from 2 to 3, and this is what we return. The output is this Mississippi and Mississippi, which has this is keyword as prefix. Okay. So these are some useful links about suffix arrays and suffix trees. I posted lecture slides on the website, so you can take a look at these if you're interested in suffix trees. Now, we are going to start talking about multiple sequences. So this brings us to this new topic, which actually we are going to spend more than 2 weeks in this. What we are going to see is we are going to see patterns and profiles. We are going to see profile hidden Markov models, and we are going to see talk about the multiple alignment problem, which is MP complete. We are going to see some heuristics that are proposed to solve this problem. Okay, so this is the outline of what we are going to see within these 2 weeks. Now, why do we consider multiple sequences? So this slide explains why we do that. Okay, why? Because usually known protein sequences, when you find the function of a protein sequence, you find similar proteins in multiple species. Even within the same species, you can find similar function genes. And these kind of similar sequences, they make up a protein family, a family of sequences. So they may have certain similarities. They are not exactly similar sequences. When you have a family of sequences with the same function, for example, the globin family, or a certain GPCR class A family. GPCR is G protein coupled receptor proteins, which interact with membrane embedded proteins. Okay, so these are receptors. Okay, they can these are membrane embedded proteins which can get some extracellular signals and interact with intracellular proteins to propagate a certain signal inside the cell. So they have, for example, if you look at these GPCRs in a certain class, they may have similar sequences. Because all these proteins, they share some things. One of them is they have 7 transmembrane helices embedded within the lipid membrane. So in the membrane of the cell, there are these transmembrane domains. And in all these GPCRs, there are 7 of them. So this is something that they share. However, they also have a lot of differences. Their loop regions at the end are very different because they can sense different receptors. They can interact with different G proteins. So it's a family of sequences, but they have similarities and differences. And it's also important to answer questions like this. When we have a new protein, which is a novel protein that we do not know anything about its function, to predict this function, it may be wise to compare it with this family. Okay, I mean, pairwise sequence comparison is not a good thing here. It's not very wise to do, because if we want to pairwise align to all the family members, and some of the family members is going to think that this sequence is not member of this family. Some of them are going to think it's very close to this family. So everybody is going to have a different say in this sequence by pairwise comparison. We would very much like to have a mechanism to just have a protein family represented by some kind of a model, okay, a protein family. So a set of sequences, we need to have a representation for a set of sequences which have the similarities of that family, which have the similar characteristics of the family represented. Also, the differences also have to be taken into account in that representation. And we would very much like to compare a single sequence against a family of sequences and say whether this new sequence belongs to that family. And in most cases, we don't have a single family. There are different families of protein sequences. And we ask this question by comparing our new sequence to multiple families. And which family it belongs to is decided by at the end of this comparison. And also these kind of family representation, these similarities between sequences that belong to the same family show us important regions of the proteins, okay, which are conserved. These conserved regions which are shared between each member protein sequence that belongs to that family means that if it's a conserved region, it means that there's something that is important going on in that region. It can be used as a drug target, or it can be used to understand the more functional detail of the family. Okay, so this is these are the main reasons we study protein sequences as families, related protein sequences. We organize them. So another main reason is that this is important for organization of the protein space. We have millions of protein sequences. If you just put them in a database one after another, unorganized, it will be difficult to make use of, interpret this data. So a hierarchical organization of this database is needed. Very similar proteins put together in families. Maybe there are families that are like each other. They may be put into superfamilies, which is a set of families. So we may have a hierarchical organization like this, which will help us to organize and understand the protein sequence space. Similar things also happen, and similar organization also needed in structural space as well. We need to organize the protein structural space to understand different structural folds and their relationships and etc. Now, we are going to start with the most intuitive, simple representation. If we are given a set of sequences, and if we are given a multiple alignment of them, so somebody used some algorithm, or maybe by hand, put these multiple sequences one after another and align them. Why do we do alignment? To reveal their similar regions, dissimilar regions. So to understand, we cannot just put every sequence on top of each other without any alignment, because they are of different lengths. So when we are analyzing these sequence of the family of protein sequences, the first thing we need to do is align them. Align their sequences to reveal common regions, to reveal non-common regions. So this is what this alignment shows. This alignment shows that, for example, the central region is shared. I mean, the central region is the same. The CTA, TC is exactly, actually we have some differences here in the middle. So in two of the proteins, this T are mutated to C. So this CTA, however, the CTA region is exactly the same. And in this region, we have some small differences. And in some, this protein, which is the longest one in this family, has certain insertions. The CCT is inserted into this first sequence. And the last G is also common to all of them. So there are common single nucleotides common to all family members. There are non-common parts, inserted parts, deleted parts. This part is deleted in the second sequence. And this is another one that is inserted. But it has something missing also. Even the longest one may have some missing nucleotides as well. The first position is missing in this family. Now, if this is our family, if somebody gives us this multiple sequence alignment, what do we do? How can we represent this sequence with a single representation? What we can do is, okay, we can say that the easiest way, this profile representation is the easiest way to do this, to represent this family. We can say that, okay, this family consists of this many locations, okay, the positions in the multiple sequence alignment. The first position, second position, third position. So each position, how many positions in total we have? Like 5, 10, 13. So these 13 positions, we can summarize what happens in each of these 13 positions. The first position, for example, what we can say is in the 20% of the sequences, this first position is missing. We have a gap at that point. Okay, since, I mean, it's 20%, it seems like a high percentage, but since we have only 5 sequences in this family, it means that there's one sequence that is missing this first position. But these analyses are usually done on much larger set of sequences, like 100 sequences. And you align 100 sequences in a family, so these percentages will be more meaningful in that case. And, but it's still, even with this small number of sequences, this profile tells us something. So what is this profile? This profile is an array of 13. So it's again like a sequence itself, okay? This profile itself is like a sequence. But at each sequence position, it's a sequence of length 13, but at each sequence position, we don't have a single letter. Instead, we have a distribution of letters and gaps. Okay, so this is a simple profile representation in which at each position in the sequence, we have a distribution of nucleotides here. Here the distribution is just A, it's all A and nothing else. In the second position, if another sequence wants to belong to this family, it has to have an A in the second position, okay, because all the family members in this family has a A in their second position. Or do we really want to have such a strict rule? You may also think about that. I mean, do we want to have such a strict rule? If maybe, are we really sure that we covered all the protein families and maybe somebody else comes up another set of 7 sequences which belongs to this, I mean, which contains these 5 of them, but some additional 2 sequences which also they are sure that they belong to this family, and maybe these 2 sequences, one of them may be missing that A in the second position, okay? So if we are not sure that we cover everything about this family, we may not be certain that, okay, this A in the second position, imagine you have a sequence with exactly the same sequence, okay, but as T at the second position, okay, like CTG, CTAT, CGC, and GG, okay? It's 90% similar to the last sequence, but just missing an A at the second position. So would you want to rule that sequence out of this family or not? Probably not. So we are going to see how we can extend this profile representation to account for such differences. But this, you understand what the profile is, right? It's just at every position, we have a distribution of amino acids. So this is our profile, simple profile representation. Now, we want to, given a profile, this is what we want to do now. How can we compare a new sequence to a profile? So our algorithms, dynamic programming, the sequence alignment algorithm, we know how to compare 2 sequences by building up a table, right? So this dynamic programming algorithm, we know how to compare 2 sequences, CATA, I mean, we can compare 2 sequences. Now we need to extend those algorithms to enable us to compare a sequence against a profile. So in this, if this is the profile, for example, it's not going to have an A. It's going to say that, okay, I'm 80% A, 20% T. Okay, the sequence is going to look, the profile is going to look something like this, and we need to be able to extend our algorithm to compare a sequence against a profile. How similar is this sequence to this profile? And this extension turns out to be a very simple extension. We just need to say that. We need to just combine we need to do a weighted average of our match and mismatch scores for this position. For example, if you want to match this A with this profile position, okay, all you need to do is, okay, treat this character as an 80% A and 20% T. So the score that you are going to get by matching these 2 things should be 80% of the match score and 20% of the mismatch score. Okay, this is all you need to do to do the. I mean, you don't need to do any modification in your original dynamic programming algorithm. All you need to do is to modify the match-mismatch scores. How do you compute the match score and mismatch score? The only if you modify this, we can actually do this alignment. Okay. Now this brings us to the topic of position-specific scoring matrices. This tells us okay, we can actually, in order to compute these on the fly, okay, for example, 80% A, 20% T. Imagine these were amino acids. We need to go ahead and look at the blossom table and see what is the score of matching A with an A, what is the penalty of matching A with a T. We need to look at these from the blossom table each time and see what is the score of matching this position with A. Okay, and we need to compute this on the fly for each amino acid. Each position is going to have a different score for each of these matches or mismatches. And this can, what position-specific scoring matrices provides us is that they give us pre-computed scoring values for each position. So what does it mean? It means that you don't need to go ahead and look at the blossom table. For each position here, we are going to have 20 entries. So it's going to be like this. We had these 13 positions in the alignment. Okay, the 13 positions in the alignment. And for each position, we list, again, let's assume these are amino acids, okay? A, C, D, E, F, G, it's like this. So we are going to have 20 rows. And for this position, we pre-compute what is the score of aligning a C at this position. Okay, and this can be computed by just weighted average of aligning C with, for example, if this was, again, let me change this to C, 80% A and 20% C, it's going to be the mismatch between C and A plus the match between C and C. And we are going to multiply this one with 0.8 and this one with 0.2. Okay, so it's going to be a weighted average of this match between C and a C and a mismatch between C and an A. And this is, we can compute this, whatever our sequence is, single sequences, we can compute this for all possible 20 matches. And this one, what we are going to have here in this table is called a position-specific scoring matrix. Why is it called position-specific? Because at each position in this family, for position 4, for position 13, we have a different score for aligning its specific amino acid to that position. For example, to match an A to position 4 may have a higher score, but it may have a lower score to align the same amino acid A to position 13. So these can also be computed on the fly like this while you are doing the dynamic programming alignment, but position score specific matrix are like pre-computed profile. It's not only a profile, it shows us how each profile position likes to match a specific amino acid. In all these 20 amino acids in the nature, how does this profile location wants to match to these amino acids in nature, 20 amino acids in nature? So we can obtain position-specific scoring matrix if we have a multiple alignment of the family of sequences. And we are going to see some different techniques to obtain these position-specific scoring matrix. And the first thing we can do is, this is the thing that I described here, which is just described more formally, and it's nothing more than that. So here, suppose we have a multiple alignment of n sequences. So in a position, it says that in this family of protein sequences, at a specific position, we have n amino acids. Okay, at a specific position, we have aligned n amino acids. If we have n sequences in that family at a specific position, for example, position I have n amino acids. Now, to obtain the values of the position-specific scoring matrix, so position-specific scoring matrix is a matrix like this. So, and here the indices means this. What is the score of aligning the youth position to an amino acid B? Okay, this is for example the fourth position to a specific amino acid B. So we can use the technique we have used to align a sequence to a profile and use the frequencies of amino acids at each column as coefficients. In other words, what we can do, the coefficients is that, so this B here is by counting in this position how many times, so this FUB is just 0.2 and 0.8 that I computed in the previous example. For other amino acids, it's 0.0, okay? For two of them, for A and C, so if A occurs this many times, for example, if A occurs 80% of the case and C occurs 20% of the case, all the other 18 amino acids, they are 0.0 because this NUB is 0 for them. So for all B, for all possible amino acids, you compute this frequency by simply dividing their number of occurrence in that column to the total number of sequences. So this is what FF is. And this MUA is just what I had written here, okay? It is the score of aligning an amino acid A with the B. So it's, for example, if you want to find the score of aligning E with this AC, okay? So if you want to, for example, find the score of at this first position, the first position index is 1, and we want the score of aligning E to this first position. This is what this these indices indicate, okay? So the first position, align an E to that first position. What is your score? Our score is going to be, for all residue types B, so for all 20 residues, actually for 18 of them, we are going to have 0 value for this FUB, okay? So FUB shows the frequency of occurrence of that amino acid in that position 1. And we know that only A and C occurs in this position, so 18 of the amino acids are going to have 0.0 as FUB. So all I have, so they're not going to contribute anything to the score since they are 0. This FUB is 0. This summation term, they can be cancelled out. So this, what we are going to have at the end is 0.2 times score of aligning E with A, sorry, C, 0.2 was the C, and plus 0.8 times score of aligning E with an A. Okay, so this is what we do. So for all the possible, so because 0.2 of this position 1 is C, and 0.8 of that position is A. So if you want to find out what is the score of aligning E with that position, you just do a weighted average of this. So this is the simple, this MUA is our first simple position-specific scoring matrix. But people have found, researchers have found problems with this position-specific scoring matrix. One of the problems is that the score is linear, in terms of, I mean, since we have these frequencies directly multiplied with the scores, if there's a certain amino acid that occurs twice in that column, in that position, its effect is going to be twice. Okay, so I mean the frequencies are directly multiplied with these scores. So the effect is linearly. We see the effect linearly in the position specific scoring matrix. Sometimes this may not be a very good thing. Maybe we may need to make this effect more. If an amino acid occurs twice, instead of using just a linearly weighted average, we may use maybe quadratically weighted average to increase the effect of those, because that amino acid occurs twice may mean a lot of things to us. I mean, it may, in terms of the scoring, we may need the effect to be more than twice. So the extensions that I'm going to talk about to this MUA matrix, the basic matrix that is computed, is based on these observations, based on these needs to change the linear effect and to make it nonlinear. So this is, I mean, the equations start looking scarier, but do not get intimidated by these equations. Their main purpose is just to make the frequency, make the previous linear. So it's like this. If the frequency, this is our frequency, when frequency is increasing, the effect of that amino acid in the score is also linear. Okay, this is our effect on the score, effect on score. It's also linear. So the main purpose of changing these equations is that to make it something else, maybe to make it something like this or something like this based on your biological motivation, or maybe if you're happy with this linear effect, you may use the original PSSM matrix. But obviously, there was a need for this extension, and this is what they do. And also, there's another thing that they do here. And this is, so the first motivation was the preferred residues, we want to give extra weight. The preferred residues means that if that residue occurs twice more compared to another residue of that higher frequency, we need to increase their weights in this. So instead of 0.2, 0.8, we want to, for example, do 0.9, 0.1. This is what we want to do. Even if this occurs, it's 20%, the majority of this is A, okay? To account for this, we want to give more weight to this A. And one way to do is to use this, the log of the natural logarithm. So this is ln. So the natural logarithm of these frequencies, okay, and add these. So the natural logarithm of the frequency, so and this, we define something, a new frequency, f prime, as the count divided by m plus 1, okay, so that we make sure this does not be, this is always less than 1, okay, we don't want ln 0 here. Therefore, we make this f prime something smaller than 1. It's always, if this n is equal to capital N, in the extreme case, the small n can be equal to capital N if all the amino acids are the same in that column. If all the amino acids are same in that column, this is going to be n number of sequences you have divided by n plus 1. And this one is going to be a very small number. Okay, this one is going to be a very small, so it's going to be ln 1 minus 0.999 something. It's going to be something like that. So a very small number, and this is actually, it's going to be even smaller. So it's, let's see what will happen if it's 0. If it's 0, it's going to be ln 1. What is ln 1? 0. Okay, so it's going to be, and if it's not 0, so ln 1 is 0. If this is 0.01, for example, it's going to be a negative number. Okay, it's going to be minus something. If this difference 1 minus this, if this f prime was 99%, okay? If it turns out that our amino acid occurs 99% at that column. So we already established the other case, other extreme case. If it doesn't occur at all, this one is going to be ln 1, 0. So its contribution is going to be nothing. If an amino acid does not occur in that column, this one f prime ub is going to be 0. And ln 1 minus 0, we are going to have ln 1. And that part, this part is going to be 0. If it occurs in high percentage, and this f prime ub is 0.99, if it's 0.99, we are going to have 1 minus 0.99, 0.01. It's going to be a negative number, a negative large number. And it's always going to be negative because since this f prime is a number between 0 and 1, it cannot be 1, but it's something 0 and a number less than 1. Therefore, the thing here is going to be always less than 1. So we are always, this upper part is always going to be a negative number. This is also a negative number. So what we get here is something positive. And if it's a small negative number, what we are going to get at the end is, just compare these two things. Compare 0.1 with 0.0001, okay? If you take the ln, this is a much larger negative number. If it was base 10, for example, this would be minus 4, this would be minus 1, okay? So minus 4 divided by some constant, and minus 1 divided by some negative constant. These are both negative constants. So ln 1 over m plus 1 is a constant number. It doesn't change from amino acid to amino acid. It's going to be a constant, but it's, again, constant smaller than 1. So it's going to be, so this shows us that if this f prime is closer to 0.999, you are going to have larger, you are going to have a higher effect in this frequency, in this weighted average. Okay, so this is how we just make this linear effect, non-linear effect by using, and this is not the only extension. You could have proposed something else. This formula is not something that is the rule of thumb. This is just one heuristic. Okay, one heuristic to make the position-specific matrix and give a non-linear effect. You could have used a polynomial function there. Okay, you could have said, okay, let me use a quadratic, for example, f square. Use the direct f computed frequencies and just take its square and use it as in the weighted average. Okay, you could. This is just one of the heuristics that can be done to compute the position-specific matrix. And another technique that is used, actually, let's have a break and continue after the break with these other position-specific scoring matrices.